Thank you, everyone. Okay, so you're just checking if online they can hear me, right? So I'll just go ahead and assume that you can hear me online. And so, yes, so this is a proceedings talk. So there's a URL at the bottom of the screen. It will be on every slide. That's where you have the slides. So, you know, if you don't need to take pictures or anything, you can just go there and you have everything that I'm presenting. I'll also say so that the majority of this work was not done by me. So Xiaojun Pan is the first author, and he's a student co-supervised by me and Xingming Zhao. So, you know, I just get to present Xiaojun's work. But let me see. Oh. All right. So let me just introduce a little bit. So the metagenomics binning problem. So this is what, you know, in real life. So the bacteria, you know, have these nice circular genomes, you know, and they live in these communities where here you see, you know, there's a yellow one there. There's many copies of the green genome. There's the magenta one, etc. And so this is what we have in the real world. And then we go and we rudely take a sample and sequence them, and out of the sequencer we get these very short reads. So we get these millions of them. They're very short. So the original genome is in the order of a few megabase pairs, and then the reads are in the order of a few hundred. And the problem is here I've colored them, but we don't actually know which original genome they come from, right? So the first thing we do is we assemble them and we make these longer chunks that we call contigs, and we are going to assume that the contigs, and this is really important, that the contigs are pure. So here, there at the bottom, we have one example where we have a chimeric contig that's magenta and green, but actually with another student, we've shown that this does happen, but it's very, very rare. So from our point of view, we're actually going to assume that the contigs we have are themselves pure. And now the problem of binning is trying to group the contigs so that ideally we have these groups that are represent, you know, some of the genomes that were in the original sample. So from this point of view, we're looking at it as a clustering problem. Right? We have a set of things, contigs, and we want to group them into bins, and we use features. And you know, this is now a 10-year-old field, and the features that we use are were well established in the field, which is we use abundance. So if we have a lot of copies of the purple genome, then contigs from that genome will also be present in many copies. And this works especially well if you have multiple samples. And then we use genomic features, in particular, K-mer features and formers have been shown empirically to work really, really well. An alternative approach is to classify contigs. And so we can have contigs. So instead of clustering, we're seeing this as classification, and we just assign them a label, right? And if this worked, then there would be no point in the binning, right? Because we would just classify everything, and we would be done. But there's two problems with this. So one is real samples are going to have species that we don't even know about. So here, like they're represented by the green one. So our classifier doesn't even know about green because it's the first time we've sequenced that species. But even species that we know about, so bacteria and like humans, they have this immense genomic variation. So there might be, so the classifier actually does know about this magenta species, but there might be whole chunks of the particular strain in your sample that are not in the database. So this is why classification doesn't solve the problem, right? So the original idea behind SemiBin is to then, and actually this depends a little bit on your metagenome, but typically the majority of them are not going to get a classification. And the initial idea behind the first version of SemiBin, which I'm going to be calling now SemiBin2, was to then combine these, try not to throw the whole baby with the bathwater, but say, okay, we can do classification a little bit, and that is informative. But how do we exploit this to then also get better binnings? And so the idea is that we are going to use this. We're going to take our contigs, and we are going to learn the relationships between pairs of contigs. And there's two types of relationships that we can learn. So one is we cannot link relationships. So this means that we're telling the system, the machine learning system, that these two contigs should not come from the same genome, right? And in this case, we were in the first version. What we were doing is we were saying if two contigs get a different classification and it's a confident different classification, then they shouldn't be linked, right? And on the other direction, we took these big contigs, the bigger ones, and as I said, we were assuming that they're all pure, right? So we artificially break them up in the middle, and now we have two smaller contigs, but because we are assuming that they're pure, we know that these two artificially created contigs should come from the same one. So in that case, we're saying this is must link. Then we calculate the features as I showed before, and then we learn this neural network that's learning an embedding. And I'll just get into this a little bit in the next slide, where the embedding tries to represent these relationships, and then we proceed with some standard sort of clustering on the embedded space. So now this is actually the more detailed slide on the math. So this is the idea of the embedding is, so we have our input. So it's our vectors of size 136, because that if you have a former and if you collapse the reverse complements, this is how many different formers you have. And then what we want is we want to go from this x to x prime and learn this function that now this is the embedding now in a 100-dimensional space, such that things that must be linked are closer together and things that cannot be linked are pushed apart. So we define a loss function. So the loss function, if things must be linked, is the distance, because we want the further away they are, that's bad, so that's a loss. Whereas the other loss is, and here to avoid things from exploding, we have to set up an upper limit, but we want things to be as far apart up to 1, right? And so this is, and then the total loss is the loss of the must link plus the loss of the cannot link, and then this is now very standard. We use just a very standard neural network learned with the very standard techniques, and this we have a reference there for so this part of the work we didn't come up with this. All right, so does it work? All right, I'm going to jump directly to the punchline. Does it give you good results? And so we compared it against other binning methods, and we used both Kami data and also real data. So there's an issue in real data is we don't know what the answer is supposed to be. So, but we're going to estimate it, and we use these two tools, CheckM and Junk, that give us an estimate of how good our data or output is. And we used 4 different data types. So we have human gut, a non-human gut sample from dogs, and then marine and soil representing environmental data. And here's the punchline. And so these are the 4 environments, and you can see that. So here this is the number of high quality bins, so the high quality genomes that we obtain. And so green is SemiBin. So if you take away, so green bar higher is good. So green bar higher. So and we can actually do this either one sample at a time, or we can combine all of the samples. So the data sets have about 100 samples each, and those are these ones here, where generally we get better results, and VAM also gets better results. But comparing like to like, green bar is bigger. Right? So this is great. And then here you can see each dot is an individual sample. So even at individual samples, we're almost always better, not just in aggregate. So this was great. This worked very well. And now want to look a little bit in what's happening. So as I said, we do these 2 steps. So we learn this embedding, and then we do the clustering. Maybe we use just a really good clustering method, right? And maybe, or maybe we have all of these steps, and maybe the embedding doesn't really help that much. It's just the fact the embedding is just transferring our classified classification information, our supervision. And so we did these ablation studies. So no semi means we don't learn any embedding, or we just hard code. We just at the clustering step we just hard code the inputs. And either the must link, the cannot links, or both. But still the full pipeline. So green bar higher, right? So the semi-supervised step is really learning something, and it's also because there was this supervision, we were also worried maybe we're just recovering more variants of species that are already known, which would already be great, right? It would already be a good advanced. But in fact, yes, because, just show you this again. So remember, there were these two issues. So one is there's completely new species, the green one, and there's these new variants, the magenta one. Maybe we're just doing really well on recovering things that are already known, but no. So we recover both more known and unknown genomes compared to MetaBat2, and we recover more, so more diversity, at all the different levels, so across the different habitats. So this is what we were, and so the results look really good. But this machine learning stuff, and also doing the classification, this was resource intensive, right? Compared to some of the alternative methods were much faster, and so we were using MM62, which is actually really, really excellent, but we were paying a computational cost, right? And so the first, and also the memory requirements were quite high. So this meant that for a computational lab like us was not a big deal, but you couldn't run this on your average laptop, right, or average consumer laptop. So we decided to try. The first idea we had is, okay, so but if we're learning a model, maybe instead of learning a model each time, we can just learn a model one time and then reuse it, or at least run a model on a few samples and then just reuse it for for all of them. And this worked really well, actually. Especially, it's better if you learn a model from multiple samples at once, but it plateaus. So so maybe you get about 10 samples, and that's about as good as you're going to get. And then if you have 1,000 samples, you can just reapply that one model one time. So you're still paying a large computational cost, but you're paying it one time instead of paying it for each sample. So this was really nice. We even tested that this worked, across data sets. So, for example, looking here, human gut, different human gut data sets. So we had 2 data sets from Germany and 1 data set from an African population. And so if both within Germany, which is 2 different data sets, but should be a very similar population, and across the world, it still works really well if we pre-train and then transfer the model. And then we also make, we made these models available for 10 different habitats, and then Florian actually contributed one for chicken. So this is where we were when we had SemiBin2, which was really, really good results. Slow to train, requires a lot of memory, but for single sample binning, so when we're using one sample at a time, we can reuse models. So we were quite happy with this. But there were 2 issues. So one is, I said, and I sort of said it in passing, that you can use multiple samples, the abundance of multiple samples. And then the problem is pre-training models doesn't really work, because we have to pre-train a model for when you have 11 samples, and then one for when you have 12 samples, and one when you have 30. And it just doesn't. We're just not going to provide pre-trained models for n from 1 to 1,000. So that wasn't working. And then the other thing, which is a completely unrelated topic, is we were seeing more and more long read data coming out. We are actually also starting to use long read data in our own lab. So we wanted to explore that as well. And so that's now the impetus for SemiBin2. So that one step at a time. So in SemiBin1, so we were using this self-supervised mode, which means from must links, we were just exploiting the structure of the problem, and then cannot links, we were using this reference taxonomy, and this is actually where the name SemiBin comes from. And then now in version 2, we decided to try what seems like a terrible idea, but we're just going to randomly pick contigs. So we're still doing the breaking up for must link, but to generate cannot links, we just pick 2 contigs at random, and we say the probability that they come from the same genome is really low, so we're going to say that they cannot be linked. And there's 2 advantages to this. So first of all, it's really fast. It's much faster to do random sampling than to do taxonomic classification. And the second one, which is also important, is that there's no database biases. So, taxonomic classification of MM6 is really, really excellent, but we cannot ask the tool to give us classifications for species it has never seen. And so this means that we are only ever going to be covering a part of the problem space, right? On the other hand, if we're doing this randomly, sometimes we're going to get it wrong, right? Sometimes we're just going to get unlucky and pick 2 pairs that actually do come from the same genome. So now it ends up being a bit of an empirical question of, which one works best. And fortunately, so the results look like this. So you can see again, so green bar is what we're looking at, SemiBin2, and you can see that you can either the bars overlap between the 2 versions, or if anything, the new version is slightly better. And if we look in this, so this is the same real data set that we had before, again, green bar higher. So green bar higher is good. So this worked really well. And then the other thing, as I said, wanted to do was to extend it to long reads. So, and the big difference between short reads and long reads is that short reads are short and long reads are long. And this sounds really, really dumb, but what it means is that the statistics of your problem just look, so these are 3 samples that happen to be sequenced with 3 different technologies. So the classical Illumina and then 2 long read technologies. And as you can see, so they have roughly the same number of base pairs as input, but in the case of the Illumina, you end up with contigs that are about, in the low thousands of base pairs long, and you have hundreds of thousands of them. And in the case of the long reads, you end up with contigs that are in the hundreds of thousands of base pairs, but you have many fewer of them. So this means 2 things. So first of all, you're clustering a lot fewer contigs as input, and your output clusters are sometimes really small. Like you can have, so this is the average, right? So, but you can have contigs that are practically a full genome, right? And actually, sometimes you even get the full genome, or you're binning and it's 2 contigs or 3 contigs, and that's your bin because your contigs are much longer. So, therefore, the clustering approach that we were using, it works. You garbage in, garbage out, right? You give the algorithm something, it will compute and it will output something, but it was just not designed for that, right? And so what we do here now is, so we have still the same, still the same initial pipeline, now with a new way of learning the cannot links, and then at the clustering step we use the old clustering method if we're doing short reads, and now we have a new clustering approach if we're doing long reads, which is a clustering approach based on TV scan that is able to give you these very small clusters as well. Again, the proof of the eating is in the pudding, and so we checked, and to be fair, a lot of the tools we're checking against, they were also not designed for long reads. So I want to acknowledge that we're misusing some of this, but there's just not. This is still very new for us and for everybody else. So, but again, a human gut, non-human gut, and then an environmental sample or active, kind of environmental-ish. And then green bar is us, green bar is higher, right? And here also note that, so one of the things, and this applies to us, but actually I think applies to others in the field, is we're using marker genes to guide the clustering. And then here we checked CheckM, which is the tool that we're using. In its first version, it's also using marker genes to evaluate the quality. So there's a bit of a circularity issues, right? And I think maybe, so to avoid this, actually, we were kind of lucky because in the meanwhile, there's a new version, CheckM2, which does not rely on this fixed set of marker genes. So here we switched to using CheckM2 to avoid, I think it's a slightly fairer comparison, but in fact, the results are very similar with both tools. All right, so I'm almost done. I just want to, but I'll leave you. So this one final anecdote. This is a very minor technical detail. I don't think it even made it into the paper, but it's sort of, so as I said, we use this single copy marker genes, right? And so, and this is Vapnik. I think maybe most of you are too young. So this was a very important person in machine learning. And develop a lot of machine learning theory, and he had this thing about do not solve as an intermediate step a problem that's harder than what you're actually trying to do, which is actually something that's really easy to do. So because in this case, we wanted to find these marker genes in our context, and so we were using Prodigal, which is an excellent at finding genes, and then we would take these predicted genes, and then we'd feed them to this HMM, and then we would only keep, the very small fraction that was a hit in our HMMs, right? And so we tried something doing, the dumbest possible ORF finder, which is we just scan, and then we see a start. We start, that's an ORF, and then it goes on until we see a stop, and that's an ORF, right? And as long as we see enough nucleotides, we say, okay, this is an ORF, right? There's none of this fancy modeling. Not unexpectedly, so that's our naive one. That's Prodigal, and this is just how much time it takes. So we are much, much faster because we're not doing any computations. We're doing the dumbest possible thing. And the result is also that we end up with about 10 times more predicted ORFs. Probably 90% of these are false positives. But then we feed them all to the HMM, so it takes a little bit longer to process all of our ORFs, but it's still faster, and the results are almost identical, right? And it's not even clear. I've never seen a sample where then the binning result changes, because we predict all of these rubbish ORFs, but if they're rubbish, they're not gonna match the HMM, right? So the end result is still really good. So this is a very technical detail. I don't think it's in the paper even, but I thought it was interesting. All right, so now, I just want to mention, like, if you don't care about anything that I've just said about all of this machine learning and the internals, and you just have data and you just want to get good mags, then we do also care about SemiBin as a tool. So it's available through the command line, but now and also because people have contributed interfaces to other workflow engines. We made a lot of documentation. We have YouTube tutorials, and if you have any problems, any issues, really just get in touch. We try to be as responsive. So we really do want people to not just think that we have cool machine learning stuff, but actually use them for your mags and for your biology. So even if you don't care about anything I've said about the internals, it is, and we try to make it a good tool. All right. So just to summarize, going back to the beginning. So SemiBin2 was this idea of using semi-supervised learning for doing the binning. And it worked really, really well. And it was combining a bit of self-supervised and reference training. It worked across a bunch of habitats, but it was very slow. So actually we ended up, this was called SemiBin because it was semi-supervised, but we ended up throwing away the semi-supervised version. And now it's fully self-supervised. So we skipped a whole bunch of computation, and the results are maybe even slightly better at a fraction of the computational cost. So now we've also added support for long reads, and it works, just across a bunch of on simulated data from Kami, on real data across a bunch of different habitats, and in the paper we've tested 10 different habitats. It just works really well, consistently works really well. So we'll keep developing, and now we're applying it to a bunch of metagenomics data sets. So again, I want to say that Xiaojun Pan was the person leading most of the work. So I want to thank also the people who are using and who are contributing bug reports, and Florian in particular. He's even contributed some models, but Berenice also contributed the Galaxy wrapper. So we want to thank all the people there. I want to thank the people who paid for it, and I want to thank you for your attention.
Thank you very much for this great presentation. So we have time for a couple of questions.
Yeah, thank you. Great talk. It seems like you could pretty easily apply this to transcript data too. Do you see any issues with that?
In what exact? So you
like metatranscriptomes.
Metatranscriptome. I mean, some of the ideas, yes. So the thing with metatranscriptomes is that, right, it's so even within a cell, you're going to have some transcripts that are higher abundance than others. So I think you could do this to try to recover some other type of associations, right? You recover things that are transcribed together, which is maybe a really cool thing to find operon-like structures. So maybe it's a very cool thing to do. Yes, I had never thought about that until you asked this question 5 seconds ago. So I'm sort of responding off the cuff, but maybe it's a very interesting thing to do. But yeah.
Thanks.
Very interesting talk. As a person that has used binning tools extensively, it's quite interesting to see a tool that outperforms everything else out there. So I'm just interested in how do you assess quality? Did you consider contamination as well?
Yeah. So we outsource that to CheckM and Junk.
Mm-hmm.
So right, and so CheckM. So actually now we have both CheckM1 and CheckM2. So that they so in the cases that we've looked, they don't agree 100%, but the message that we do better stays the same. Mm-hmm. And Junk checks for contamination with slightly different way of checking for contamination.
Mm-hmm.
So
also single copy genes.
No. So Junk looks at this would be a different talk, but so what Junk does, it looks for contigs that seem to be out of place, that don't match the rest of the contigs of the bin, but based on all the genes, not just single copy ones.
Very cool. Thank you very much.
Thank you for the very informative talk. I have two detailed questions about the method.
Yep.
First, because you use learning,
yep,
how does the number of contigs and the contig length affect the performance? That's the first question. The second question is, maybe I missed this in your talk. Did you do some cross-domain validation? For example, all the training data are from human, but you apply this on other
yep,
like animals.
Yep. So first, the first question is, so it's not exactly the number of contigs, but the number of samples. What we see is, so if we increase the number of samples, we see it plateau. So as we're learning, 2 is better than 1, but when it gets to 5, it sort of seems to, or 5, 10, it sort of seems to plateau. And then as to your second question, I think I have it as one of my extra slides. So thank you for asking, because I got to show it. Yeah. So here is we've tried learning a model on one habitat and then transferring it to the other. And it's interesting that you always do better if you train and evaluate on the same habitat, even if it's a different data set. And it's also interesting that similar-ish habitats, so for example, if you transfer between, so here you can see between the mammalian guts, it's actually still okay, and if you transfer from soil to it's more different. So I'm not exactly sure what's going on. Maybe the taxonomic composition introduces, but yes. So we can transfer across habitats. You pay a cost. Yeah.
Thank you.
Thanks a lot, Luis. I have a question regarding the marker genes. I assume you are using 40 marker genes, and might it be the issue from soil to gut, for example, since we don't know the exact marker genes because of unknown species, that might introduce this missing translation, let's say?
Yeah, so we actually use a data set it's 107 single copy genes. So there is something, and we've actually discussed it internally just recently, which is maybe it's not appropriate for archaea. So I think if you're in the bacterial domain, you're probably still fine. And since most of our data sets are very enriched for bacteria, we were fine. But this was actually a user who emailed me and said, well, my data sets have a lot of archaea, so maybe but that's at this level of going from archaea to bacteria. If we're within bacteria, they should be universal, right? Universal in the biological sense, which means 90+%, but not universal in the mathematical sense.
Thank you. So we have one more question online. Maybe you can see that directly.
Oh, sorry. How do I? Sorry, I got it. Yeah. Now that I'm also going to read it for the audience here. Now that long reads are catered for in SemiBin2, and more errors encountered in long reads like nanopore. Does this fully supervised approach that is faster work as well for these long reads? Yes, I mean, and to my surprise, somewhat to some extent, it actually, so the supervision, the model, you can learn a model on short reads and apply it on long reads, and it works well. So it seems that, also here we're not applying it directly to the reads. This is also important. We're applying to the contigs that have been assembled, and in some cases they've been polished. They've been polished, so the contigs themselves should have fewer errors than the long reads initially had. But then it does seem that the models, it doesn't seem to make a big difference at that stage of the process.